Okay, welcome to chapter 3. So, chapter 3 is really cool. This is actually one of those chapters that we use a lot, not only in class, but also in the lab. So, chapter 3 is all about microscopy and how we observe microbes in the lab. Okay, so we're going to talk about some basics, and then we're going to talk about some optics, and then we'll get into some details about what we can see underneath the microscope. So our chapter 3 objectives is we need to be able to explain how light microscopy reveals information about pathogens that we isolate from patients. We are going to look at a bunch of different types of microbial cells use under the microscope. We're going to be able to look at an image from a microscope and we'll be able to determine what type of microscopy was used. And then we are going to need to be able to use bright field microscopy, fluorescent microscopy, and electron microscopy to explain the information we get from certain micrographs. So micrographs are what we call the pictures we get from the microscope. So let's start with a case study. So chapter 3's case study is called Pathogen in a Can. So this case study takes place in the good old state of Oklahoma. So we're going to follow Mariana and Julio, who are two kids aged 8 and 11 respectively. Now, this story takes place in the middle of the afternoon. Both of Mariana and Julio come to their mom and they complain about not being able to see correctly. Like they're having trouble seeing things. And their faces were starting to turn numb. Okay, so when they come and start talking to their mom, it's about an hour after they had eaten lunch. For lunch, their mom had cooked them a couple of hot dogs and then topped them with a commercially canned hot dog chili sauce. Because who doesn't like a good chili dog? Now, understandably, their mom got a little bit freaked out when they said that their faces were going numb. So their mom took them to the urgent care, where they saw the PA that was on call. Now, the first thing the PA did is to take the kids' vital signs. Now, both of the kids' vital signs were actually normal, okay, but there was some weird inconsistencies. One of the things that was weird was that their pupils were fixed. So when you shine a light in your eye, your pupil, or the black part, will expand and contract. Now, if you look here in this picture, right here, you see how big their pupils are? They stayed that big and didn't change size when the PA shined the light in their eyes. Okay, when it came to the numbness in their face, the numbness or the weakness was symmetrical, meaning that it was equally weak on the left and on the right. Like they tried to have them smile, they had tried to have them blink their eyes, and they saw the same response on both sides. Now, while they were seeing the PA, unfortunately, their weakness in their face progressed to full-on paralysis. Okay, and then they started to have a difficulty breathing. So their lungs are starting to be paralyzed. The PA understandably freaks out as well and says we have to get them to the hospital. This is bad. So they call an ambulance. But both Mariana and Julio are transferred to the nearest medical center. Okay, so once they get arrived at the ER, they are taken back for a bunch of tests. So they have some blood drawn. One of the tests that they use their blood for is what is called a specific monoclonal antibody test. So antibodies are immune molecules that are used to target specific molecular shapes or specific proteins. Now, one of the tests that they have run, because of the paralysis in their faces and the fixed pupils and their difficulty breathing, was the antibody test for botulinum toxin. Okay, so what this antibody does, it has a fluorescent dye. Fluorescent means it glows. In fact, we call the part that glows in the dye the fluorophore. So they have this glowing dye, and when the antibody attaches to the specific protein or shape that it's for, when we look at it underneath a fluorescent microscope, we'll see it glowing. So they performed this test, and unfortunately, they saw this underneath the microscope. So we can see where the fluorescent dye is. Okay, so this showed that they had this toxin in their bloodstream. Okay, because they saw this and because of their symptoms, both Mariana and Julio were diagnosed with botulism. So botulism is an illness that is characterized by paralysis. Now, Botulism is associated with foods that have been contaminated with the bacterium Clostridium botulinum. Okay, so this is not great. And in the course of being in the ER, Mariana and Julio are admitted, and mainly because of their difficulty breathing, and they have more and more trouble breathing until finally the hospital puts both of them on a mechanical ventilator, which is going to be a machine that breathes for them. Okay, so after both Mariana and Julio were diagnosed with botulism, they were they were prescribed a cocktail of antibiotics to help treat the infection. So after a few days of being on the antibiotics, the paralysis in their lungs was reversed, so both of them were able to come off the ventilators. But it took quite a few treatments of antibiotics and some physical therapy before both kids were fully recovered. Now, when they were diagnosed with botulism when they came into the hospital, this launched an investigation with the health department. So both the hot dog factory and the canning factory were tested for traces of both the bacterium and the toxin. So the hot dog factory came back clean, there was nothing there. But unfortunately for the canning factory, they did find evidence of both the bacteria and the toxins, and the toxin in cans of food. In fact, they found 6 cans that tested positive for the botulinum toxin. So they closed it down. They recalled that chili sauce. Okay, so when they were testing these cans, one of the things they did is they looked at under the microscope at the contents of these cans. So when you look underneath the microscope, when you see Clostridium botulinum, this is what it's going to look like. Well, let me turn my. Okay, this is what it looks like. Okay, so this one is actually in the process of dividing and making 2 cells. But what we have here is we have a spade-shaped bacterium. The part I want you to focus on here is this pink part. Now, one of the things Clostridium botulinum can do is it can form an endospore. Now, if you remember back to John Tyndall's experiment back with the S-curve flask, one of the problems he was seeing when he was trying to disprove spontaneous generation, it was that he was getting growth in his meat broth despite sterilizing it with heat. And we now know that he had a bacterium that would make endospores. So this does the same thing. So when a bacterium forms endospores, what's going on is it's preparing itself for harsh conditions and to survive those harsh conditions. So it's going to put almost all the genetic material in that endospore. And when that endospore is formed, it's going to be both heat resistant and chemical resistant, and it won't dry out. So it's going to allow the bacterium to survive until there's better conditions. Now, when we can food, if you don't use a pressure cooker and just use heat like a lot of canning factories, because this is an endospore, just boiling it won't kill the endospore. We have to have that pressure element as well. So this is how we have, oh, sorry. This is one of the reasons why this bacterium and this toxin is found mostly in food contamination. Now, canned food isn't the only thing that can be contaminated with this toxin. Clostridium botulinum can actually get into honey in the beehives. And this is actually one of the reasons why babies aren't allowed to have honey until after a certain age, because of this possibility of having it be contaminated with this bacterium. Now, if a baby is fed the honey that is contaminated and that toxin gets into their bloodstream, they go on to undergo full paralysis. So they get what's called floppy baby syndrome. So they can't control any of their muscles, their lungs seize up, their heart stops beating. It's actually pretty terrible. But thankfully, no one else in this case study was affected like Mariana and Julio, and they were able to pull the contaminated food off the grocery store shelves. Now, that is the case study. I have a question for you. What do you think we use the botulism toxin for? Okay, this actually it's a little weird, guys. What does botulism toxin sound like? Well, if you said Botox, you'd be correct. The treatment Botox is literally we're taking refined and purified botulinum toxin and we're injecting it into the places in your face where you have wrinkles to paralyze those muscles and smooth your wrinkles out. Like we're literally using a paralytic to do that. I won't judge. If people want Botox, they can have Botox. It just seems a little extreme to me. I don't think my beauty practices need to go quite that far. I'm actually quite proud of what wrinkles I have. I worked hard for them. So yeah, it's kind of a weird use, and it's a weird way to show how ingenious humanity can be sometimes. But we can see here, after the injection, this lady had the wrinkles. After she paralyzes those muscles, those wrinkles are gone for well, until the Botox wears off. Okay, so that brings us to 3.1, and we're going to all talk about how we observe microbes. Okay, so we're going to talk about microscopy and some of the basics of it. So we're going to talk about two parameters called magnification and resolution, and then we're going to talk about some of the different kinds of microscopy and what they really mean. Now, why do we care about microscopy? What do we need to know about it? One of the things in both the medical field and in science, if we can make a visual observation, that speeds up the process. Okay, and it's actually an effective way to start going towards a diagnosis. Now, we have, let me get my mouse here. Okay, so on the left, we have these two vials. So these vials are both filled with CSF, or cerebral spinal fluid. Okay, so our CSF is one of our sterile sites on our body, meaning there should be no microbial growth in our CSF fluid. So our cerebral spinal fluid actually helps cushion our brains. So if that's where it's found, is around the meningeal areas. Don't quote me on that. Okay, so the vial on the left is clear. This is how your CSF should always look. Now, the vial on the right, see how cloudy it is? This is actually not infection. This is actually white blood cells in the CSF fluid that have formed and congregated because of an infection. Now, with your cerebral spinal fluid, when it's cloudy like this is not good news. Because it's found mostly around your brain and in your nervous system, this could indicate that you have a brain infection. Now, we see this test come back a lot with people who have been diagnosed with meningitis, and we'll talk more about that in a later chapter. Microscopy also allows us to get a view of the bacteria. Okay, so we can see here we have these spherical single bacterium in this sample. So we can look at a tissue sample or a blood sample underneath the microscope, and we can look at the shape of the bacteria, and we can use stains to help us figure out what type of bacteria they are. Okay, so this would be quicker than waiting, say, for DNA sequencing or some of our growth tests. So we can start a treatment that's going to get rid of this microbe, hopefully faster than if we waited for all the other tests. Now, it doesn't always ring true, or sometimes there's way too many bacteria that have the same arrangement and shape, but it's a place to start. Okay, so we need to define microscopic. So we know that the definition of a microbe is a organism that you have to view underneath a microscope. So microscopic is literally means viewing the tiny or seeing the small. Okay, so anything that's microscopic requires a microscope to be seen. So microscopes use two parameters, or two things by which they work. The first is magnification. So we define magnification as an increase to the apparent size of an object. So you can think about it as zooming in. Okay, the other parameter is resolution. So resolution is the smallest distance by which two objects can be separated and still be able to be distinguished. Okay, so what this means is how far away can you get the objects? Okay, or how close can you get the objects and still tell the two objects apart from each other? Now, let's talk about the human eye, because our human eye kind of acts like a microscope. So our eyes have very high resolution. Okay, so the human eye has a resolution of around 150 micrometers. Okay, our eyes are set up with, let me get my mouse here. We have the iris, we have the cornea, we have the pupil, we have the lens. Okay, we have our retina, and we have our optic nerve. So here in the retina area, it's made up of what we call photoreceptors. So photoreceptors are just proteins in the back of our eye that catch light. We've talked about them back in physical science. Now, if we get light that comes in directly from the eye and hits straight back to the back of our eye, we have really great eyesight. Things are crisp and clear in the images. Well, one of the reasons this is because straight back is where most of the photoreceptors are. Now, if you look at the side of the eye and in your peripheral vision, it's not going to look as clear. It's going to be a little bit fuzzy or just not as resolved. Okay, it doesn't have as great of resolution as if you were staring at something straight on. And that's because the number of photoreceptors catching light is going to be a lot less when you're looking out the side of your eye. Now, one thing that I need to mention is when we use light to view objects, so whether this be our eye or be the microscopes we're going to be using in class, so we have this guy that we're looking at, and that light is going to bounce off of him and hit our eye. The lens in our eye is going to gather it. What happens is when our lens gathers it, it's actually going to invert the image. So we're actually looking at the guy upside down, or the image of the guy is actually upside down when it hits the back of our eyeball. But our brain is programmed to flip that up, so we're looking at him as if he's right side up. Okay, this happens in the microscope as well, except our brains don't flip it like it does with our eyes. Okay. Now, have you ever heard the term that this person has an eagle eye? Well, this is actually quite fun because eagles have 8 times the resolution in their eyes that humans do. So if we have a resolution of 150 micrometers, that means that eagles have a resolution in their eyes about 1200 micrometers, which will allow them to be high in the sky and be able to pick out those mice in the fields when they're hunting. So they can stay high and then swoop down and catch their prey. Okay, so let's talk about high versus low resolution. Obviously, the higher the resolution, the more that we're going to be able to see. So if we look in these pictures are of the Grand Tetons. The picture on the left is a high resolution image. So you're able to see the snow, you're able to see the crags and mountains. We can look at the tree line and distinguish individual trees. We can see the grass, we can see what's growing in the lake. But what's really cool too is that we can see the reflection of both the mountains and the trees in the lake and be able to distinguish them from each other. Now, if we look at the low resolution image, we can kind of make out that there's mountains back there, maybe a little bit of snow. We look in the tree line, but we can't really distinguish individual trees unless they grow above the tree line. And while we can see that there is a reflection, we really can't resolve it or see it clearly like we can in the high image picture. Now, one of the things that we have to remember is magnification and resolution work together. Okay, so they're interconnected. Now, you can think of how you take a picture on your phone. So our phone cameras are rated by pixels. Pixels are the resolution of our cameras in our phones. So if we take a picture and we zoom in on a specific part of that picture, we can zoom in, but there comes a point where when we're magnifying that picture, that it goes blurry and pixelated. When we get to that part, we have gone past the resolution of our camera, and it can no longer distinguish between different objects, and which is why it becomes blurry. So now that we've talked about resolution, we need to talk about microbial size. So with our resolution, exactly how small can we see? Now, when we talk about the size of microbes, cell size differs between the different microbes, but we need to talk about some common units used in microbiology. So we have millimeters. So 1 millimeter is equal to 1/1000th of a meter, or 10 to the minus 3 meters. We use micrometers. Micrometers are probably one of the ones that we use the most. Microbiology, micrometer, kind of makes sense. 1 micrometer is 1/1,000,000th of a meter, or 10 to the minus 6 meters. We use nanometers, which are 1 times 10 to the minus 9th meters, and we use picometers, which is 1 times 10 to the minus 12th meters. Now, we do sometimes use another unit that is between nanometer and picometer, and that is the angstrom. Angstroms are 1 times 10 to the minus 10th meters. So we use angstroms a lot in chemistry, which is why they've kind of moved over to microbiology a little bit, because some of the microscopes that we use, especially some of the electron microscopes, can actually image atoms. So that's how that came to be. Now, let's look at some examples of some different microbes that we could see underneath the microscope and some that we cannot. Okay, so this first guy, the biggest one here, is Trypanosoma brucei. This guy is 33 micrometers. Oh, let's talk about one more thing before we move on. When we abbreviate micrometer, we use this weird looking U. This is actually the Greek letter mu. So this is how we abbreviate that. Okay, so brucei is 33 micrometers. Brucei is 4 times bigger than our red blood cells, which are about 8 micrometers across. Now, if we talk about bacteria, because bacteria are all smaller than our red blood cells, the biggest bacteria that we know of is Bacillus megaterium, which is about 4 micrometers long. Okay, good old E. coli is about 2.5 micrometers, and Staphylococcus aureus, which can cause staph infections in MRSA, is 0.9 micrometers. Now, there's things that are smaller than bacteria, like viruses. So the herpes virus that can cause your cold sores is about 0.2 micrometers long. Okay, depending on what we need to see and their sizes is going to determine what type of microscopy we use. Now let's talk about the different types of microscopy. So we have light microscopes. Okay, this is going to use light as the optics, or the way that we view the images. So there's going to be a light source on the bottom, and because there's a light source on the bottom, the background is pretty clear and bright. So in order to see our samples, we use special stains to add contrast. We have phase contrast microscopy. This is going to use a special filter that's going to allow us to see both stained and unstained samples. And we'll see an example of this of all of these actually on the next couple slides, where we look at some micrographs. We have good old fluorescent microscopy. This is what we saw in the earlier case study. Okay, this is when we use a stain that has a fluorescent dye, and we use different wavelengths of light to excite the fluorescent dye so we can see certain microbes underneath the microscope. Okay, light microscopes, we also sometimes call these bright field microscopy or bright field microscopes. Phase contrast microscopes and fluorescent microscopes all use some version of light in how they work. Now, the next couple of microscopes are a whole different category. These are electron microscopes. So instead of using light, we use a beam of electrons to image things. Okay, so we have 2 types that we're going to talk about. There are a few more types than these ones, especially the one that we use to image atoms. Okay, so we have scanning electron microscopy, or SEM. This one's pretty cool because it gives us a 3D image of the microbes that we're looking at, and it has high resolution, so it gives us a lot of surface details, so we can see what the outside of the microbe looks like. Then we have transmission electron microscopy. This is TEM. And this is going to be very similar to a light micrograph in how it looks, except we're going to have extremely high resolution so we can see a lot more details. In fact, we can see so many details, we're going to actually start to see the parts or the organelles in the cell. Okay, so the next couple of slides are a set of tables and images from our book. And this kind of illustrates the differences between the different forms of microscopy. So in each of these micrographs, we have the same microbe, and it's Giardia lamblia. Okay, so we actually have this microbe in the river, down in the river bottoms. If we took a fresh sample, more often than not, we'd actually get some Giardia in there. Now, with light microscopy, this is where we use a stain. Okay, so this is going to be the lowest resolution type of microscopy out of all the ones we've talked about. Okay, so we're going to look here at Giardia. We're going to use they use a methylene blue stain in this micrograph. So when we use stains, the stain's going to adhere to different parts of the cell and not others. So some areas are going to appear darker, some are lighter, and some areas aren't going to have any stain at all. And what parts of the cell get stained depends on the stain that you're using. So they're using methylene blue here. I don't think we use methylene blue in the lab this semester. Don't quote me on that. But we use crystal violet, safranin, and iodine a lot. Okay, so the methylene blue is allowing us to see the nuclei of Giardia, and we can kind of see where the flagella start. Okay, so phase contrast, this is going to use a filter. So what it's going to do is it's going to darken the background, and we can see these transparent microbes pop up. So this is going to give us an image that is going to look somewhat 3D. So we can start to see different details on the surface of this microbe. So like we can see, kind of see where the nuclei are. We can see the flagella. Now we can use the dark background like it is here, but there's also a light background that we can use with samples that are stained. Now the fluorescent microscopy, we saw this in the case study. Okay, so we're going to use a fluorescent molecule to label different parts of the cell, and then we'll be able to view it underneath the fluorescent microscope. Okay, so different dyes are going to be attached to different antibodies, which are going to label different parts. So in this picture, our fluorescent molecule is going to be bound to the outside of the cell and to the flagella. So that's what we see here in the image. Okay, we also use fluorescent microscopy for a bunch of other things besides diagnoses and to view things. We actually use it to count living cells in a sample. Common terminology is green means it's alive and red is dead. So if we were looking at a bunch of different bacteria under a microscope and we were using fluorescent microscopy to see which ones are alive and which ones are dead, we look and count the green ones to see which ones are living. Okay, so that is our light microscopy. We are now going to go through and look at the electron microscopes. So remember, for SEM, our scanning electron microscopy, we're going to see a lot of details on the outside of the microbe. Okay, so we're going to get the most three-dimensional image we can get with microscopy. And just a reminder, as we're going through from light microscopes up, our resolution's increasing, which is why we can see different details as we move up in microscopes. Okay, so we have a lot more definition here. We can see these wrinkly areas, we can see whatever this is, and we can see the flagella. So it's a pretty cool image, actually. And then the final type of microscopy we're going to look at is transmission electron microscopy. Okay, so our TEM. Now this is the one that has such high resolution, we're going to start to see things inside the cell. Okay, so it's going to look the most like a light micrograph with more details. So we can see the nuclei, we can see the base of the flagella, we can see the cytoplasm and some of the vesicles that are inside the cell, so that's pretty cool. So when we talk about microscopy and the different sizes of things that we can see, we really need to look at the scale of what we're actually seeing. So on this slide, we have a scale. It ranges from 1 millimeter up to 1 nanometer, which is the same as 10 angstroms. So whenever you see this A with a halo, this is an angstrom unit. Okay, so we have a bunch of different micrographs of things at different sizes. So we have different types of resolution. So let's talk about our human eye. Our human eye can view things from about 1 millimeter or bigger down to about 150 micrometers. Okay, so our eyes can't see a lot of very small things. Now, light microscopy is going to overlap with what area our eyes cover, but it's going to go about from 1 millimeter down to about 500 nanometers. Okay, so in image B here, this is a common light micrograph. This is actually something that we will be seeing when we do start to make slides in the lab. This is good old E. coli. This is what it looks like underneath the microscope. Now, we're going to get into our electron microscopy, our electron microscopes. So a scanning electron goes from about 1 millimeter down to about 5 nanometers or so. Okay, so we can see here in this image in C, this is what E. coli looks underneath like using electron microscopy. So we can start to see some surface detail images. This is TEM, so it's not going to be as much of a 3D image as if we had used SEM, but we can start to see some details. Okay, we can see the cell wall, we can see the stuff on the appendages on the outside, but look right here, we see these really dark circles. Okay, these little dark lumps. These are actually pretty cool. Now, TEM is going to go from about 15 micrometers down to 5 nanometers. Okay, so this is going to allow us to look inside the cell, look at this different cell parts. So if you remember these little dark images here, well, this is what they look like. So these little dark circles here are the ribosomes. So they're what makes the proteins. So if we look carefully here, we can see the mRNA, and we can see the ribosomes attached to the mRNA, and we can see the protein strands, or the protein sequences that those ribosomes are making. Isn't that pretty cool? Now, if we need to go smaller than these 5 nanometers, we're going to use not microscopy at all. We're actually going to use X-ray crystallography. Now, if you remember correctly, this is the same method that Rosalind Franklin used to image DNA, so we could discover that it was a double helix. Okay, so we're going to crystallize our sample, and we're going to bombard it with X-rays, and the X-rays are going to bounce off the individual atoms in the sample. And then we're going to take all the data that we get, and we're going to use a computer and computer software to model it. So this is actually a computer image, which is why it looks a little bit weird. It looks kind of globby and kind of weird. This is actually what the ribosome looks like. So we can see the two subunits, the large subunit and the small subunit, which is pretty cool. Now, we need to talk about viruses. What kind of microscope can we use to see viruses? The answer is we use electron microscopy to see viruses. So we won't be seeing any in our labs. Our light microscopes aren't good enough to see objects that small. We don't have the resolution. So if somebody tells you at our school in the biology lab, oh, I saw a virus today, they are lying to you. We can't see viruses with our microscopes. Okay, so we are going to talk about the last thing in this lecture, and that is the shapes of bacteria that we can see underneath the microscope. We need to know these, guys. You need to memorize these shapes, because this is going to come in handy on the next exam, and this is going to show up several times in the lab. So this is something we need to know. So the 3 common shapes of bacteria are rods. We call this, oh, let me get my pointer. Okay, so rods are referred to as bacilli. Okay, so E. coli is a rod-shaped or bacillus-shaped microbe. We have spirochetes. They're going to look like corkscrews, and they're corkscrew shaped. We've seen spirochetes before. So if you remember the case study about the stockbroker from Chicago that got syphilis, syphilis is a spirochete. And then finally, the last shape of bacteria that we have is the cocci shape. So these are the round spherical microbes. Okay, so these top images are from a light microscope with stains, and then these bottom ones are from a scanning electron microscope. Now, we need to talk about one more thing. Electrons aren't colored. So when we take a micrograph from an electron microscope, our image is going to be black and white. So when you start to see these electron micrographs colored, we actually do that after we collect the micrograph from the microscope. Okay, so this is not what is actually being seen underneath the microscope.